Welcome back to Office Hours Unplugged, where we chat with the most prominent researchers and PIs here in McMaster. Today we have Dr. Cameron Currie joining us. It's a pleasure to have you on the show. Could you please give a little introduction to the people who may be a little unfamiliar with the biochemistry department?
Yeah, hi again. Thanks for having me. I'm a faculty member in the biochemistry department, biomedical sciences, and biochemistry department, and here at McMaster in the Faculty of Health Sciences.
Awesome.
That's nice. How did your journey in academia start off? I mean, take us from the very beginning here.
Yeah, so I like probably many of your listeners, entered university not sure what I wanted to do. There was a range of things I had in mind, like physiotherapist or maybe going to medical school. And so I specialized in biology. And it was in my third year that I started taking ecological oriented classes on a little bit of environmental science, evolutionary biology. And I became really interested in that topic, but I was still uncertain of how that translates into a career. And then I was really fortunate to get to know a master's student in one of my higher level classes who was working in a lab that was hiring undergrads for the summer to do research, and suggested I apply for the job. And I think he helped me a little bit get that job. And I started doing research, and I really was fascinated with the idea of working on something that no one's, like getting an answer to something that no one's ever figured out yet, or, teasing apart something about the natural world. A lot of the time in undergrad we get told what's known. And so it's pretty exciting to be working on stuff that's unknown. And it's a little, at that time it was, less common to be exposed to research than say now at McMaster. And so that's how I got excited about research and discovery. And then the specific topic that we work on in my lab relates a lot to biocomplexity and biodiversity, and how biocomplexity and biodiversity is generated over long periods of time. And so within that realm, I became very interested in species interactions, especially mutualisms, these interactions where two organisms benefit each other. And many of these associations are obligate, where they each depend on each other for survival. And so those were systems that I got very excited about as into doing a master's. And then at that point, I still wasn't sure where that led. And then learned about, then I've got exposed to PhD level research and this stuff. And as an undergrad and master's student, I was at the University of Alberta in Edmonton, and was fortunate to join a lab at the University of Toronto for my PhD, and that's where I started working on the mutualism between ants and fungus they grow for food, which we do a lot of work on in my lab.
Yeah.
So prior to that placement, were you ever thinking about working with ants, or was that kind of like your first exposure to how ants and fungus work together?
Yeah, that's a great question. I've been described as the ant man, so I get that question quite a bit, and people are like, "Oh, did you play with ants when you were young?" And the answer to that is no. I, like lots of Canadians, I played a lot of street hockey and ice hockey when I was young, and was not really aware of this fascinating world of ants. And so it was the first exposure to ants. And what really drew me to leaf-cutting ants, fungus-growing ants, is their obligate mutualism with fungus they grow for food. So these ants have some of the most amazing biology. They grow these fungus gardens, so they use plant material to culture fungus, and then they eat the fungus for food. And so in a way they're performing a natural example of agriculture like humans do. And we grow plants for food, they actually grow fungus for food. So and so that was my first exposure to ants.
Wow, I mean, that's your lab is very broad in the topics and things that you study. I mean, how is that something you're drawn to not really be constricted to a specific field?
Yeah, it's a little bit of following, what we get excited about, thinking about new opportunities. If you look at it from the external outer view, it can look like, oh wow, they're really doing lots of different things. But the common foundation that ties it all together is being interested in the role microbes play in mediating the biology of largely animal hosts, and specifically a lot of the work is, do these microbes produce antimicrobials that mediate disease dynamics of these hosts? And so we're doing projects right now in the human nasal microbiome, which sounds very different than fungus-growing ants, but the conceptual framing is very similar in the sense of a lot of our work on fungus-growing ants involves studying these bacteria the ants maintain on their bodies that produce antimicrobials that protect their fungus garden. And because the ants are obligately dependent on their fungus for food, and their fungal crops are subject to fungal infections and diseases, the ants have evolved to use these bacteria to produce antifungals to protect their fungus garden. So it's really an interesting another parallel to humans. As I said, there's a parallel of human agriculture, but the ants not only evolved to be the first farmers in the natural world long before us. They also evolved to use bacteria for production of antibiotics. The bacteria the ants use to protect their fungus garden are the same from the same group of bacteria that we get most of our antibiotics we use pharmaceutically. And so, as I was saying, ants use bacteria to combat fungal infections. So we've then been working on the nasal microbiome, looking at are there bacteria that are beneficial bacteria in our nasal system that might be producing antimicrobials, antivirals that mediate respiratory infections.
Yeah, well, that's it's quite the rabbit hole.
Yes.
You start with the ecology, and then you just keep going where the science leads you.
Yeah.
I think that's a very beautiful thing.
Yeah, it's a fun approach too, in the sense of because we're open to using our conceptual framing into new systems. It keeps it also fresh, and it's great for training of undergrads and grad students in the sense of we've had undergrad researchers who've worked on totally new systems that no one else in the lab has worked on, and so that works well for some grad students who want to kind of carve a new path and a new route in the lab. So it's exciting.
So was this kind of fascination with this relationship and the bacteria and all these interesting phenomena what led you to create the Currie lab? Like, was that your original inspiration? And like, did you also think about doing like becoming a postdoc and doing staying in academia?
Yeah. So when I did my PhD, I started working on fungus-growing ants, and that's where I discovered the ants have these bacteria that produce antifungals. And while doing my PhD, the idea of becoming a professor was something that I felt was attractive, like as an option. I didn't necessarily, wasn't certain, of that was going to work out as a path for me, but I really fell in love with research and discovery and the work that we do, and so the opportunity to set up my own lab was really exciting. I actually started my academic career. Well, my first academic appointment as a professor was at the University of Kansas in Lawrence, Kansas. And I was there for 3 years in the ecology and evolutionary biology department. And then I moved my lab to the University of Wisconsin in Madison and was there for over 17, 18 years. And
Wow.
moved here to McMaster, my lab here to McMaster 3 years ago. And it's been really fun because in University of Wisconsin-Madison, I went from an ecology and evolutionary biology department in Kansas to the Department of Bacteriology at Wisconsin. and that was really great for me and my students, is we were around a lot of really smart people that work on bacteria and microbiology, and so we learned a lot of detailed microbiology there, and it really facilitated a lot of the work we were doing. And then now I'm in a medical school and in the Department of Biomedical Sciences and Biochemistry, with more application perspective and exposure to different expertise, and so that's really exciting new expertise exposure for my lab members and myself, and so it kind of fits a little bit with this diverse approaches, is also changing a little bit of the environments where we learn, and that keeps us growing. And it's an exciting part of being a professor, is you're really fully in this lifelong learning kind of mode in your life.
I mean, yeah, that's quite a lot of time. I mean, what really motivated that move to McMaster? I mean, you were there for 17 years in the University of Wisconsin-Madison.
Yeah, as a Canadian, it felt like it was time to come back to Canada.
Yeah.
So that was a big part of it, and I had a lot of respect and admiration for McMaster and researchers here like Jerry Wright and Marie Elliott, and that are really squarely in my field, and Eric Brown and others, and there's an aspect of the culture that's been built here of teamwork, and so there's a lot of emphasis has been placed on developing infrastructure that's shared that can be used in core facilities on campus that provide access to resources for researchers. The CMCB, for example, has a lot of robotics in their system, and we were doing a lot in Madison, a lot of what we call co-cultures, where we look, does microbe A inhibit microbe B, and pairing thousands of combinations of these things. And we published a paper in 2019 that had 55,000 pairings conducted over 3 years by, on average, probably 5 undergrad researchers each year. So a huge amount of effort on 12-well plates, all done by hand. And in the CMCB, we can use their instrumentations and robotics to run around 3,000 pairings a week with no human effort. So the scaling of the science we can do here is really exciting. And so that's a big another part of the attractive nature of coming to McMaster.
And since making this move, did you notice that there was any, I guess, particular challenges with becoming a PI in McMaster? And is there anyone that inspired you to kind of set up your lab? Because I know Dr. Truant talked a little bit about, I think his name was Brian, who had a really great lab, and he kind of inspired Truant to make his lab. So is there anyone like that for you?
Yeah, so one of the things about being a really interdisciplinary researcher is you have very different types of techniques and expertise that go on in your lab. And so one of the challenges we have in terms of how we set up our lab is we're not a standard, there's lots of outstanding labs at McMaster that do very detailed biochemistry, molecular biology, and they use similar methods and similar equipment, and we do a little bit of that, but we do many different things. And so one of the challenges of setting up a new lab is navigating the components. But when I came here, I was setting up my third lab, and technically it was my fourth, because at Wisconsin we moved into a new building after a few years, and so I reset up my lab. So some of this was just standards, setting up, resetting up the tent and following it. We definitely get a lot of inspiration from people on campus in terms of approaches and these kinds of things. And that's a great part of being here at McMaster.
Yeah, and do you think that the collaboration here is something you've enjoyed so far? I mean, I don't know how it was at the University of Wisconsin-Madison, but I do notice that McMaster is really nice at how it brings together people with the different organizations, the sub like the IIDR and that kind of thing.
Yeah, I think the collaboration is one of those things that is easier to do internationally now than it used to be, because you can do Zoom meetings and people fly around. And so in some ways, we try to maintain some of our developed collaborations over periods of time, but definitely emerging collaborations. We're very excited to be working with Jerry Wright on thinking about the evolution of antifungal resistance across the fungal kingdom is a project we're working on. Professor Marie Elliott in the biology department, have some budding collaboration. She's an incredible expert on the genetics and biology of the genus Streptomyces, which is very closely related to the genus of bacteria that occurs on the ant that we work on. And so we're excited about working with her and collaborating with her, and that's starting up, among others. And we're really excited about those opportunities here as well.
And prior to coming to McMaster, when did you, or if there was any time when you did, realize that these antimicrobial compounds from like the ants and the fungus could be used in drug discovery?
Yeah, that's a great question. The antimicrobial discovery realm is something that I very gradually moved slowly and more solidly into, and as I said, I discovered these bacteria on the ants and showed that they're producing antifungals that mediate the disease dynamics. But at that time, we didn't know any of the chemistry, and I had a opportunity to collaborate with a really talented natural product chemist named John Clardy at Harvard Medical School, and a number of postdocs in his lab over years worked on collaborations with my lab, and we characterized a lot of new antifungals from the ant system. And when we first started, it was really like trying to understand the ant system, and then we realized that there was potential for application, and part of that work, what that work allowed us to do was to apply to the National Institute of Health for antimicrobial discovery research center grants, and we were really fortunate to get one at Wisconsin, and so we had I think over $50 million worth of research grant over the years at Wisconsin to discover new antimicrobials, and so we continue to do the very interesting basic science that we're excited about, but it allowed us to actually test the what we call efficacy, the effectiveness of these antifungals we were discovering in mouse models, and fully more fully explore them. The challenge with antibiotic discovery is there's a lot of issues with getting new chemistry. What we want is to find new compounds that have not been explored before, and when you find, they're hard to find. But we've been, our systems and our approach, we've been very successful. But then most people might view that as, oh, you have a new antibiotic, but you're very far from having what the general public would be considered an antibiotic, because there's tens of millions of dollars of preclinical studies you have to do even to get to a clinical study, which could cost as much as $500 million. And there's not a lot of resources and support. Traditionally, there's not been a lot of resources to take on that preclinical work, which has been called the Valley of Death for antimicrobial discovery. So it's I think probably hard for the public to understand. Like, people hear, oh, they've discovered a new antibiotic, and think that, that might be in the pharmacy soon for use, or in the clinic. And we've I think discovered more than 30 antifungals that are new structures that could all have potential in drug leads, but we have one of them that has moved through some of the early preclinical work that we're still excited about, but it's a very slow process to take something to the clinic.
Yeah, I mean, it usually takes years.
15 years.
15 years.
Yeah, so one of the things I learned is some of the estimates are that it takes 15 years to develop a new antimicrobial, and it takes 3 years for resistance to evolve. So not a very good ratio.
Yeah. Do you think that maybe it should be something that is there's more pressure on like expediting it maybe?
Yeah, I mean, so as humans, we are often reactionary and aren't as proactive as a collective as we would benefit from being. And so there's a recent estimate that in the coming years, death from infections, infectious disease, are going to exceed cancer deaths. Maybe by as early as 2050. And so the National Institute of Health in the U.S., which funds the most, one of the biggest, arguably historically the biggest funder by far of biomedical research, about half of their $30 to $35 billion budget goes to research related to cancer, and a very small percentage, maybe even less than 100 million, goes to antimicrobial discovery. And so if you think about all of that work that's been done on cancer and investing in cancer, and you compare what we invest in antimicrobial discovery, like that's I think summarizes well, like, the general public are donating to cancer research, but you don't have people donating to antimicrobial discovery. And I think cancer research is super important, and we have many outstanding researchers in cancer research here at McMaster, but we also need to put more emphasis on antimicrobial discovery efforts.
And have you noticed that like funding changed after COVID-19? Because I feel like that was like a big wake-up call for a lot of people to show that viruses and bacteria and infections are still very dangerous. So did you notice any changes? Like, did funding change, or did the government, as soon as COVID-19 ended, they're just like, okay, no more funding for you?
Yeah, so in the U.S., the National Institute of Health, during early days of COVID, started a program that was specific to develop antivirals that was similar to the program that we had for antimicrobials. And invested a lot of money, and I think there's been some really good research that's been done with that money. But I think there has not been as much of a shift in perspective as one would like to see in terms of recognizing and investing in this. I think there's lots of reasons to one could argue, and I hear people argue, okay, well, COVID-19 was our one in a century pandemic, and we're good for another 100 years. And if you actually track emergence of viral diseases, and you look at it on a time scale, there's no doubt that they're becoming more frequent, and there's some real reasons to be concerned. When I was teaching at the University of Wisconsin in Madison in 2014, the Ebola outbreak occurred, and you both are too young to remember this, but there was actually a patient in Texas that had caught Ebola and actually infected nurses at the hospital. And so there was it was a really interesting time to be teaching in a class that was thinking about evolution of virulence and diseases and emerging pandemics. And I'd been giving lectures talking about, is there a big pandemic coming, and talking about the theoretical side. And then that happened, and it was like, look, it's on U.S. soil.
Yeah.
There's a lot of reasons to feel like we were pretty fortunate in 2014 that didn't spread further, and so we need to be more proactive and invest in these areas.
Yeah, I mean, and then you had like 6 years after that, you had another outbreak, and then, I mean, based on what you're telling me, people haven't learned their lesson yet.
Well, yeah, and then now bird flu is a concern, but there's also Zika in there. The HIVs globally. We don't have time. SARS-CoV-1, which was really relevant in the Greater Toronto Area in the 2000s. Like there was, I lecture on this here at McMaster. We talk about evolution and virulence, and transmission is an important part of the ability of pathogens to evolve resistance. Because ultimately, if you're a pathogen that is dependent on a host, if you kill your host, you die as well. So it is maladaptive for a pathogen to be highly virulent unless it improves their transmission. What improves transmission is high density and traffic of individuals. And so our high population densities and massive international travel, like most people don't know, there's 55,000 flights in the U.S. alone every day.
Wow.
So you have millions and millions of people mixing. There's huge opportunity for transmission, which can select for increased virulence. So there's dynamics to be concerned about in terms of setting up opportunities for what would be considered, in an evolutionary sense, maladaptive diseases that spread rapidly and are highly virulent.
Yeah. And since you mentioned bird flu, I actually took a course with Dr. Leslie McNeil, and I learned about the avian flu, and it's very dangerous because I heard that it has almost a 50% fatality rate, and it's developing resistance towards all the drugs we currently have. So do you have any ideas about any opinions on that? Because I feel like that's insane. And imagine a pandemic with that kind of virus. It would make COVID-19 look like a joke.
Yeah. I'm sure you'll maybe have Matt Miller on your show or Professor McNeil and speak of that. I'll say two things. One is most people are not aware that some of the fungal infections the human face have case fatality rates of 50% as well, and the resistance is an issue. So there's very similar dynamics in fungi, although the chances of rapid transmission is different for fungal disease than viral disease. But one of the things that I think about with COVID-19 a lot is one of the things we learned during that is how vulnerable our supply chain was for feeding people and getting materials around. Super vulnerable, right? In terms of the way we operate. And if you think of SARS-CoV-2, if it had the same case fatality rate as SARS-CoV-1, very closely related, which instead of 2% or lower, and not a very high case fatality rate in our young people, but SARS-CoV-2 maybe 9, 7, 8, 9, 10%, somewhere in there. Who would go out and put groceries on shelves if they had a 1 in 10 chance of dying from getting infected from the pandemic? Who would transport food to location? Who would be we would be under a real serious issue with really breakdown of our food supply chain. And then you get in a situation where people start to go hungry, and then people are torn between hunger and safety, and then you have concerns around breakdown of society. So it is really important. So one of the reasons we're working on the human microbiome is during COVID-19, we had started working on the nasal microbiome, and we were interested in whether these commensals in the nose were mediating viral diseases, respiratory diseases, and specifically rhinovirus. And then COVID-19 hit, and we're like, well, these could be producing antivirals that have effects on coronaviruses. And we actually, from one of our commensals, got a promising antiviral hit. We were behind the development of antivirals from big companies. We kind of got a little bit stalled, and fortunately some of the other antivirals come. But we're still very interested in this, because one of the few things you can have prepared in the case of a pandemic is antivirals to lower the case fatality rate for your vulnerable, for your exposed individuals while we're developing a vaccine. And so those are one of the few tools we have in our arsenal to help us in emergence of a new viral pandemic.
Okay, and I guess just a more general question, since your lab is so interdisciplinary and there's like so many different directions you can kind of take that mutualistic relationship between ants and fungus, is there any like research that you're doing right now that is like of most interest to you or is the most promising?
Yeah, it's something, so the one of the things that I'm really newly excited about is, I think we're contributing to the community and the antimicrobial development, and there's lots of outstanding work in this realm, and I'm really proud of the contributions we're making as among many others here at McMaster, and but we've been doing some really interesting basic science on the ant that has now made a connection to climate change that has me really interested, and it's a very new avenue for us, and it relates to an observation and a publication that we published near the end of my time at the University of Wisconsin-Madison, and that is we discovered that the ants have a biomineral carbonate layer on their exoskeleton that actually acts as an armor and protects the ants. And so we did the experiments where we had ants with and without their biomineral, and we did ant wars, and we showed their armor really helped them win the wars and survive in these wars. Ant wars are an important part of survival. There's a lot of aggression between species in nature and wars, and so there's a selection that occurs to have this armor, and so we published this paper on this biomineral, and it was really interesting because it's biominerals are not present, and it was the first carbonate biomineral ever discovered in an insect system, and so that was really exciting. But we'd been thinking a lot about how this forms, and we did an experiment where we took ants and we put them in closed chambers, and we put CO2 in with the stable isotope C13. So the carbon in the CO2 was labeled C13, so we could actually track it. And the reason we did this is we were wondering, are the ants actually capturing the CO2 from their chamber? Because the carbonate is CO2 based. The integration of the carbon is CO2 based, and so we're asking the question: are the ants scrubbing CO2 from their chamber? And we're about to submit the paper and found that yes, in fact, they're scrubbing the CO2 and removing it. And in writing this paper, I've been reading a lot of the geological literature, and it's a really interesting parallel to what's called weathering in terrestrial systems in geology, where you have reactions of CO2 and water on certain types of rock that actually create limestone and dolomite and carbonate rock. And over geological time, this has actually shaped our climate. And so this is a very important sink for carbon, a super important sink. And there's lots of exciting research that's trying to promote this activity to reduce the carbon, to capture carbon from the atmosphere. And so we're really excited because this geological process occurs over millions and millions of years, and our ants over hours are scrubbing CO2 from their atmosphere. So it's a really exciting little model system for processes that are sequestering carbon into a rock. The biomineral in the ant is basically a mineral rock. And the reason this is really an important area is that if you grow more trees, that's great for capturing some carbon, but that carbon all cycles back into the ecosystem. Those
Right.
trees die, they break down. This is a limited cycling thing. If you're making rock, you're capturing this carbon long term. And so converting CO2 into rock is really an exciting area in the climate mitigation. And so this is an area that I'm currently really excited about, and we're excited about exploring, because in the natural world there's not an equivalent system where a metazoan, an animal, is capturing CO2 to make a carbonate mineral rock, and so we can learn a lot from it.
Wow, so the ants are basically mini carbon sinks, essentially.
Yeah, we think they are.
Yeah.
It's an exciting thing to think about.
Those sound very interesting.
Yeah, we're not suggesting that we're going to go out and make many ant farms to capture carbon, but if we know the mechanisms, the proteins involved that facilitate and rapidly create this conversion, that could inform on attempts in a geological sense to set up conditions to do this conversion. And so that's the application we're thinking.
And have you noticed, like, if some ants take up more carbon than others, and can you selectively breed for those ants?
Yeah, so the within an individual ant colony, they're all sisters, and so they're all very closely related. And so the process and the complexity of an ant colony is much more of based on their social structure. So there's a queen, and there's different workers of different ages at different tasks, and so there's a lot of understanding, the conditions that improve and increase this carbon capture. But one thing we haven't talked about is these ants have been farming for over 65 million years. So they've already, really optimized as much as possible many of these processes. So one of the things in the antimicrobial world we're doing is we're saying, hey, humans have been using antibiotics for 80 years, when major problems with resistance. Well, ants have been doing this for 60 million years. How are they overcoming resistance and remaining effective? And so this is a natural system to do this. And so in the analogy of the carbon capture, we can say, okay, well, ants have to deal with toxic CO2 accumulation. How they learn to mediate that in this conversion. And what can we learn about this? And another way of thinking about it is, if we studied ants in the late 19th century, early 20th century, before antibiotics were discovered, and really dug into understanding how they maintain their gardens in the face of disease, one of the things you discover is they're using antibiotics, and be like, oh, well, maybe those are useful for us. Obviously, we're well behind in that regard, but there could be things like that the natural world inspires technologically in terms of digging into these things.
Fascinating.
All right. Well, we are running out of time, unfortunately. I would love to continue this conversation. This all sounds very interesting. So this has been Dr. Cameron Currie. Very exciting to hear about his work at McMaster, and we'll see you next time on Office Hours Unplugged. Thank you, Dr. Currie.
Thank you.